I just wanted to introduce our next speaker. I have known for some time. She's been an instrumental part of my thinking in terms of what happens with molecules as they move through the biosphere and how important it is for us to understand that movement and tracking. I think the science used to model and measure is such a critical place where technology plays an important role for our society and our community. So science to measure, science to model, maybe instead of manipulate, but just really understand what's happening. And you've elucidated so many things with your work. And I'm going to let you, Gabby Black, who is a PhD candidate in molecular biology from UC Davis. And her doctoral project is focused on actually tracking and implementing DNA through the point of consumer products to biosolids, and then, as you know, many biosolids are put back on ag lands. And so how are our consumer products affecting the health of our soils? And Gabby's instrumental in helping us really understand that. So I'm thrilled you're here today, and I really appreciate you making the time in your student schedule for this. Thank you.
Perfect. So thank you all so much for being here. I can't tell you how honored I am to actually be speaking at a Fibershed event after attending several in the last 6 or 7 years. And I'm extremely excited to get to share some of my doctoral work with you guys. It's very easy to get a little bit discouraged when you're studying toxic chemicals and how abundant they are in the material we're in contact with every day, but to come here and see people actually making a difference is so inspiring, and I'm just so excited to be a part of it. So like Rebecca said, I'm a PhD candidate at UC Davis in the Agricultural and Environmental Chemistry group, and I work with Professor Tom Young, and we do high-resolution analytical chemistry on a whole consortium of different chemicals, and the topic of my thesis is looking for chemicals that have originated from consumer products that have some kind of biological toxicity effect, and that endpoint I'm looking at is endocrine disruption, so what chemicals in consumer products can interfere with our hormone signaling processes. And then looking for those chemicals actually in sewage sludge as kind of a matrix that's at the end of the treatment train. We then apply that to the land, and sometimes it has a lot of chemicals in it. I am a huge advocate for the land application of sewage sludge. Please don't misconstrue my talk. I think I'm just part of a team that's really trying to make sure we're doing that safely. And any chemicals that end up in sewage sludge, maybe we don't need them in consumerism, and that's kind of my goal, is to identify what of those compounds are exceptionally nasty in consumer products. And so when I talk about consumer products, I'm talking about everything from fragrances to cleansers to pharmaceuticals, cosmetics, plastics, detergents, clothing, antimicrobial soaps, pesticides, and food additives and food preservatives and food packaging. I'm sure many of you in this room are familiar with some of these chemicals like azo dyes, parabens, phthalates, antibiotics, synthetic hormones, a variety of different pesticides and fragrances. And so all of these chemicals are of interest to me, and I'm looking at what of these chemicals are present in sewage sludge across the state of California, signifying their ubiquitous use in consumerism and overall persistent through varying forms of treatment trains. And I was really excited when Rebecca asked me to do this talk back in, I think, August, and I was like, oh my gosh, this works out perfectly with my timeline. And of course, the minute you have a deadline, things break down. And I apologize if this isn't like the smoothest flowing slide deck. I was working on it until late last night because when I went to run all my samples, our instrument went down for a month. And this data is like super hot off the presses. It like finished running on Wednesday. But for this talk, I was really excited to dig a little bit deeper into chemicals from textiles and clothing that we're finding in sewage sludge. And so unfortunately, wrinkle-resistant, stain-resistant, and waterproof clothing doesn't come from wrinkle-free sheep or water-resistant sheep. They're coated with chemicals, right? And so these chemicals can come off in the wash. They have a variety of different pathways on how they can get into the environment. But chemicals that I'm kind of focusing on today are perfluorinated chemicals, phthalates, and azo dyes, and then polybrominated azo dyes, which is a really interesting class of chemical that I'll talk about in a little bit. And so like I said, these chemicals, and so sewage sludge is this great kind of matrix that's highly concentrated. Anything you use in your everyday life that could potentially get down a drain somewhere has the potential to make it to a wastewater treatment plant. And wastewater treatment plants are designed to expose chemicals to a really robust chemical and biological treatment process. But there are so many organic contaminants in consumerism that it's absolutely impossible to require wastewater treatment plants to effectively remove every single one. What I'm more interested in is what chemicals can we remove that are causing issues. And so if you're like me, when I started my PhD, I had no idea how wastewater treatment worked. But everything that comes down the drain from a household or manufacturing goes into these sedimentation tanks. These sedimentation tanks let the solids fall to the bottom, the water advances into secondary treatment, clarification and disinfection, and then ultimately discharged into rivers and streams. The sludge, however, kind of builds up in the sedimentation tanks, so a lot of sewage comes in and a lot of sludge is concentrated before it's actually dredged out for secondary sludge treatment via either anaerobic digestion, aerobic composting, and then is ultimately applied to the landscape as part of the nutrient cycle, which I think is extremely vital. Right now we treat human waste as this kind of hazardous material, taboo material to use, but it's actually, we have this huge break in the nutrient loop, right? We grow food, we eat the food, and then we get rid of all those nutrients because we're afraid of it, and for good reason, but I think that there's room to improve on this system. And so any chemical that ultimately goes down the drain persists its intended use in consumerism, persists the transport to and treatment within the wastewater treatment plant, is cause for potential environmental concern because it's going onto the landscape. In the world of textiles, please don't worry about all the words in this diagram if you can read them. I definitely can't. But chemicals in textiles have multiple ways of getting into the environment. The primary one is we wear clothes, we wash them, the washing machine drains, goes into the wastewater treatment plant, so any chemicals that leach off of our clothes eventually make it to a wastewater treatment plant. These chemicals in the environment have shown to decrease the light penetration of surface waters. So, if you have a cadmium blue spill, the water is going to turn blue and the light's not going to get to the bottom to help with photosynthesis for primary productivity of aquatic grasses. There's also some sublethal health effects that have been tied to exposure of azo dyes, perfluorinated compounds, polybrominated compounds in the environment, such as growth reduction, metabolic stress, and also toxicity resulting in death. Has anyone heard of the term endocrine disruptor? I figured, okay, cool. So I'm gonna introduce a new term that you may or may not have heard, endocrine active compounds. So this is kind of the bigger picture of my dissertation, is looking for endocrine active, not necessarily endocrine disrupting compounds. So endocrine active compounds are those that mimic synthetic hormones, and they fit into a hormone receptor. So if we have a compound that looks very much like estrogen, it'll fit into the estrogen receptor, much like two puzzle pieces fitting together, and ignite some kind of cellular response. We don't know what that cellular response is. It could be nothing, but that could also be a problem, right? And so once we identify endocrine active compounds, it's kind of mimicking the EPA's endocrine disruptor screening program, where they test all new chemicals going into consumerism, and then the ones that are able to bind to that estrogen receptor are then advanced for further in vivo testing or animal testing to actually see if that hormone receptor binding complex produces an adverse health effect. And that adverse health effect has been highlighted recently in the news, mostly for the compound 17-beta-alpha-ethinyl estradiol, which is the primary ingredient in birth control pills that is being discharged with wastewater effluent, causing male fish to develop ovaries, causing reduced reproduction rates, and a whole host of other implications for environmental aquatic species. And so for the focus of my work, I'm looking at endocrine active with the interest of potentially bringing those endocrine active compounds into in vivo studies to better assess the adverse health effects associated with those interactions. Is everybody clear on that? It's really difficult, like, having the instrument down. I've been so in data mode. I'm trying not to be like a boring technical scientist. And so I'm sure many of you have heard some of the poster child compounds for endocrine disruptors like bisphenol A. These BPA-free labels are all over everything. BPA has been shown at high concentrations to interact with estrogen signaling. It's been linked with prostate and ovarian cancer, and there's a lot of evidence showing its connection with obesity and eventually type 2 diabetes upon exposure to infants and pregnant mothers. This is a really great example of good intention. So we figured out that BPA has these toxic effects. So we initiated this big withdrawal of BPA and consumerism, and we replaced it with this compound called bisphenol S, where they just substituted this dimethylated carbon up here with this sulfur dioxide carbon, and unfortunately we're now seeing that bisphenol S is just as toxic or more than BPA. Great example of good intention, great example of how there's just so much stuff that we don't know, and this is what we're trying to figure out. What can we find out before it goes into consumerism or ignite these kind of pullback of these chemicals? But it's so difficult to fully assess the compound, but great intentions. Another chemical you may have heard of that there's a lot of evidence for its potential endocrine disrupting characteristics is glyphosate, the active ingredient in Roundup. This compound, upon exposure in vivo animal testing, has shown reduced reproduction rates, reduced testosterone, and then also anti-estrogenic and anti-androgenic properties, meaning that this compound will bind to estrogen or androgen receptors and block any function. So if you can imagine, estrogen has a role, androgen has a role, and if it can't fulfill that role, that's also a problem. So it goes both ways. Sometimes they interact and stimulate a response, sometimes they interact and stop any kind of response. Some other suspected endocrine disruptors, and I'm saying suspected because it takes a boatload of evidence to actually say this is for sure an endocrine disrupting compound, but there's been evidence for aniline and other azo dyes found in textiles, perfluorinated compounds, which are found in nonstick cookware, but also in waterproof, water-resistant, stain-proof, and wrinkle-resistant clothing, phthalates, which are found pretty regularly in screen-printed t-shirts. You know how if you have an old screen-printed t-shirt, maybe it's kind of worn off? It's because the phthalates probably came off in the washing machine. Polybrominated diphenyl ethers, which are kind of a legacy class of flame retardants found in furniture foams and other fabrics. These have been phased out quite a bit and replaced with tributyl phosphate flame retardants, which we'll talk about in a little bit, and then polychlorinated biphenyls, which are present in a lot of our electronic coatings. So all of these classes of compounds have evidence suggesting their role as an endocrine disruptor. And so like I said, I'm looking at sewage sludge. I'm looking for all of these compounds from textiles in sewage sludge, meaning we wore them, we washed them, the chemicals came out in the wash, and now they're in the sludge, and that sludge is getting land applied, so what do we need to be concerned about? And so I have 14 wastewater treatment facilities in California who have graciously volunteered 1-gallon sludge samples. And all of these sludge samples have been subjected to a slight variation in primary wastewater treatment. So they come from different diverse inputs into the wastewater treatment plant. They're treated mostly the same, but there are some variations, and then the sludge is taken out, and the secondary sludge treatment is also a little bit varying. Some of these sludges are aerobically composted, some are anaerobically digested, and some of them are just laid out to sit for 5 years, and then they put them on brownfields. Some of these are bagged and sold as compost. And so the differing level of treatment is what I wanted for this study, because I want to know what chemicals are in consumerism throughout California and persist all these different kind of treatment modes. So the first part of my dissertation, which, believe it or not, took me like 2 years, was to actually figure out how to extract all these chemicals from sludge. A lot of people, I think, stay away from sludge because it is kind of a gross matrix, but once you get past the smell, it's just a really complex matrix. And so to be able to effectively pull out hormones, dyes, perfluorinated compounds, antibiotics, plastics, all in one sweep is a challenge. And so I spent like 20 months trying to figure that out. But we have something that we're calling good enough, and there's evidence to show that it is good enough. But I just wanted to put in this kind of workflow so you can kind of see the big picture. We're going to focus on this top row right here. Once I extract these compounds, we run them on this magical box, which is called a liquid chromatography quadrupole time of flight mass spectrometer. We'll call it an LCMS. What this does is, all you need to know for today is that it's a really high resolution workhorse that can just churn out really precise, really detailed data. And the data set is humongous, which is wonderful because we're able to analyze this data in so many different ways. Today I'm going to be talking about the 3 different modes of analysis that we perform, which is targeted, suspect, and non-targeted data analysis, and I'll talk about that on my next slide. But I also just wanted to kind of reiterate kind of the big picture here. So our extracts go on our LCMS and we analyze the compounds that we see, and I'll talk about this more later, but like I said, the data set is so huge, sometimes narrowing down what we want to look at is very difficult. So at the same time, we're taking those extracts and feeding them onto plates that we have cells attached to, and these cells are super rich in like estrogen receptors or super rich in glucocorticoid receptors. So when we feed our extract onto those cells, if something interacts with that receptor, we've designed these cells to then, the cellular response is designed so we can measure it. And so we can know what parts of the extract are estrogen active or glucocorticoid active. And with that activity data, we can do statistical analysis combined with this targeted, suspect, and non-targeted chemical analysis and try and figure out which compounds are responsible for that activity. Because in any extract, we could have 10,000 compounds, and it's with fractionation on our LC, we're able to continuously kind of go in this circle and narrow down which chemical is responsible for that activity. But like I said, we're just going to focus up here because that's where I am in my PhD, and maybe in a couple years when I come back, we can talk about the whole circle. So there's 3 different ways we analyze this data set. The first is a targeted screen. And as I think Donald Rumsfeld said, who was a Secretary of Defense, said that there are things that we know, there are things we know we don't know, and there are things we don't know we don't know. And so the targeted screen are the known knowns. We know these chemicals exist in science, and we know these chemicals should be in sludge. Examples of this are aniline dyes, phthalates in clothing, estradiol, and different hormones. And so this is like having a treasure map where there's an X and there's a map and there's clues, and we can just go up and look for the treasure, and we find it if it's there and we don't if it's not. It's the easiest, most simple way of data analysis. The second way is our suspect screen. So the things we know we don't know. So these are chemicals known to science, but we don't really think they should be in sludge, but we're going to look anyway. So we have a database of spectral entries from pesticides to forensic toxicants to recreational drugs that we can screen all of our sewage sludge data against. Examples of this is this 8-hydroxyefaverins, which is a metabolite of a drug, fipronil, which I guess now I'm not surprised that it's in sludge. Fipronil is the active ingredient in the flea and tick stuff people put on the back of their animals' necks. And this is like a detective, whoops, got ahead of myself there. A detective looking at a crime scene and looking at the relevant stuff, but also keeping an eye out for the stuff that maybe doesn't jump out. So our third way of analysis is this non-targeted screen. So things we don't know, which I can also compare to wedding planning. People were asking me, like, do you have everything? I was like, well, I don't know. And so examples of these chemical-wise are transformation products of chemicals that, maybe you washed your hand with an antimicrobial soap and that triclosan broke down on its way to the wastewater treatment plant. And we're only looking for triclosan, not the hydroxylated form. So this allows us to look unbiasedly at chemicals that we've never seen before. And this is like looking through a haystack at 1 million pieces of hay and trying to pick out 10 pieces of hay that are more important than the other 9 million pieces of hay. It's not even like finding a needle, because if you find it, you don't know all the time. And so I'm going to go through some of my results. I geared a lot of this data towards chemicals in textiles and clothing, which is actually, I've never focused this hard on textile chemicals before, and so I think that this is pretty interesting. So the targeted screen, I looked through the literature at what chemicals are in clothes. A lot of them are azo dyes. A lot of them are polybrominated compounds, which are much like the polybrominated diphenyl ether, flame retardants that have since been phased out of consumerism, and then perfluorinated chemicals, so the water-resistant, stain-resistant chemicals. And And what I found was actually pretty interesting. So in this paper by John Geisy in 2016, he found more mutagenic polybrominated azo dyes than polybrominated diphenyl ethers in household dust. And so I took all those polybrominated azo dyes and looked for them in my data and actually found a ton of them, which is crazy and kind of cool. And so all of these compounds that have like the Geisy or Geisy in a number means it's one of his mutagenic polybrominated azo dyes, so mutagenic dye. Among this targeted data, I found several dyes, this chloroaniline and thiodianiline. So I'm sorry, I should explain this. Different compounds I looked for are on the y-axis, and then the percent at the bottom is the percent of samples they were present in. So we have DEHP, which is bis-2-ethylhexyl phthalate, which is one of the most common plastics in clothing. And just in general, was I think in 86% of the 14 sludge samples across all replicates. So not only are we seeing plastics, we're seeing dyes, dye intermediates that are made in the production or dyeing of clothing. This is a little sticky, sorry. Flame retardants and a chemical used in the production of polyester, and then fluorotelmer alcohols, which are a waterproofing agent in clothing. And so I have to kind of forewarn you, this is all pretty preliminary. I made this graph yesterday. And normally before saying this is absolutely this compound, we do need to do some manual confirmation. I went through and manually confirmed many of these, but please don't quote me on this to anyone of importance, like the government or something, because I still need to, I'd say I'm 80% sure these are the compounds that I'm saying they are. So then moving on to the suspect screen results. We have a database of about 5,000 entries that range from common water contaminants to forensic toxicants to pesticides. And then there's probably a few hundred of just kind of random chemicals that we've come across in our lab that we have spectra for already from our mass spec. In this data, I found one compound that was present in all 14 sludge samples across all 4 replicates of each, and that was 17-alpha estradiol, which is the predominant chemical in the estrogen patch, which is prescribed for menopausal women. We saw a lot of compounds kind of at higher abundances here. The 2 bars are just different ionization modes. Don't worry too much about that, but we saw a lot of antihistamines and a lot of antifungals found in topical antifungal treatments. So yeah, there's a lot of stuff in there. There was another compound I meant to, sorry, can't really see. This TCPP is a tributyl phosphate. This is in newer flame retardants or newer materials. And so that's just an example of kind of the phased out compounds. So then on to the non-targeted analysis, the unknown unknowns are trying to differentiate which piece of hay is more important than another in a huge pile of hay. Per sample, I'm finding 15,000 entities. And to simplify the word entity, each entity could potentially be a chemical. The problem with this is that there's many naturally occurring chemicals or plant-derived chemicals in sludge, and those are usually present at much higher abundances or concentrations than the anthropogenic toxic chemicals. And so we need a way of prioritizing these 15,000 entities to go after which ones we're trying to identify, which ones we're trying to distinguish as an endocrine active or endocrine disruptive compound. So this chart is a principal component analysis of 15,000 entities in one of the samples. Each square is an entity, so just kind of a good depiction of how much stuff we're dealing with. The chemicals over here, how PCA works, the chemicals over here are a lot different than the ones over here, and any entities right next to each other are very similar. So one way to prioritize these entities or potential chemicals is to look at detection frequency. So just like I did with the target and suspect work, what chemicals are there in all of the data? So what azo dyes are in all 14 samples, what dye intermediates, what perfluorinated compounds are in all sludge samples across California. And when we run that filter, we get from 15,000 entities down to 500. 500 is still a lot, but it's more manageable. So we can then look at each entity, like this red square here is C18H30O2. It could be linoleic acid, which is an amino acid, but we'll do some more testing to confirm that. But this is how we can start narrowing them down. So we can narrow them down by nitrogen if it has a nitrogen, we can narrow it down. All azo groups have nitrogen, so that's one way of running a filter. With this work, we found tributyl phosphates, which are the newer flame retardants, like I said, ivermectin, which is a dewormer, beta blockers, sedatives, and then in all 14 samples across all 4 replicates, we found 3-amino-benzoic acid, which is a dye intermediate. So in every single sample that I have from California, there are dye intermediates in the sludge. So it's making it through the wash, through the wastewater treatment plant, and are partitioning into the sludge and being introduced in the environment. The next steps for this work would, like I said, to be measuring these endocrine active characteristics, and we do that in a way that's what's called an effects-directed analysis. And so we pair our LCMS analysis with endocrine activity or biological response. We can then churn out, hopefully, identifications for endocrine active compounds. With those endocrine active compounds, we then kind of advocate for further in vivo testing to see if they do actually exhibit some kind of adverse health effect. And I'm running a little low on time, but in conclusion, there are many chemicals from textiles that I'm finding, maybe not in 100% of sludge, but 50%, 70%, 80%. Those range from azo dyes, perfluorinated compounds, polybrominated compounds, phthalates. All of these things are making it into our environment from the clothes we wear. And so that's one of the reasons I just love Fibershed and love this event, because it's people making a difference on this issue. And so I commend all of you. That's fantastic. And with that, I just want to acknowledge my professor, Tom Young, the gracious support and funding from the Jenna and Michael King Foundation, John Wick and Peggy Rathman, the National Science Foundation, and then our stellar collaborators at Agilent Technologies. And with that, thank you very much for listening. I would love to take questions if we have time.
Yeah, there's one.
I apologize, I have 2 questions. I'll throw them really quickly.
Okay.
One question is, in terms of the laundry landscape system, which is being implemented throughout the North Bay, I understand that water is not being treated, but it's also not being concentrated. So it might be outside your window of research. Do you have any thoughts on whether those endocrine disruptors or endocrine active compounds are more or less of an issue in that kind of system? The other question is in terms of using fungus as a metabolism agent in further processing biosecurity, and whether that might have an influence on the amount of endocrine active compounds present in the end product.
I'm going to answer the second one first. Absolutely. Biology dictates chemistry. Microbial processes, fungal processes transform these compounds into potentially inert compounds on an endocrine scale. So yes, absolutely. I think any kind of biological material you introduce or organism you introduce has the potential to change the chemical composition significantly. I'm not familiar with the laundry to landscape.
It's just gray water use. Taking
Okay.
a laundry, a washing machine to drain it directly onto your landscape.
Yeah. And so you were asking if I think there's a difference in the concentrated versus not concentrated?
Exactly, yeah.
It's kind of an old school train of thought to think that dilution is the solution to pollution. I think that that's no longer a strong belief, so I think concentrated or not concentrated, I'm not sure that it would make a difference, but I do know that a lot of these endocrine disrupting compounds are effective at causing disruption in the parts per billion, parts per trillion range. So I don't really, with my way of thinking, I don't really think it would make that big of a difference, but don't quote me on that. Okay.
I have 2 quick ones as well. So we're working on microfiber, and so one question is that cool magic box identifying physical microfibers and of what type?
Our magic box only looks at organic contaminants, so chemicals. There are other magic Agilent boxes that can look at different things.
Yeah, we use their FTIR on board.
Okay, yeah.
And so my other question is, do you have a feel for when the disconnection happens? And do you think that the breaking up of clothes hastens the disconnection of these chemicals into the water. So like they have to disconnect from your clothes. Do you think it happens in the wash, or do you think it accelerates as it gets to wastewater treatment, or any feel for that?
I'm afraid I don't know the dynamics of clothing and absorption and desorption of chemicals well enough to give you a really educated answer, but I would guess that the first few washes are probably much more concentrated in these chemicals than the latter washes, and that's just because people tell me I need to always wash my clothes before I wear them. And you see that the dyes come out earlier with like a new pair of blue jeans. They're gonna dye your white shirt you wash them with on the first wash, but maybe not on the 10th wash. And that's my very uneducated, naive answer. I'm sorry, I can't give you a better one.
That's awesome. That's really helpful. Your work is super fascinating.
Thank you. Thank you. Thank you guys so much.